मेरे कुछ दोस्त भेजते हैं तेरा अकाउंट क्या है? नहीं मैं स्टार्ट ये नहीं मेरा दोस्त कौन है? जो फिर ऐसे बना मैं नहीं सर देखना ही इसका नहीं मेरे पास एक आईडी है इसका नाम बता मेरे दोस्त ने बनाया होगा 100% नहीं तुमने बताया कभी नहीं आया अभी
Never charge.
मेरे एक दोस्त है उसपे 465 फॉलोअर्स
मेरी बिल्डिंग काफी सारी
अरे क्यों बोल रहा हूँ 465 हाँ हाँ मेरे बिल्डिंग में बनते हैं तेरे कितने मेरे 6 मैंने तेरे 20 मैंने प्रोफाइल फोटो भी नहीं डाला है मैंने कोई भी
सिर्फ देखिए
हाँ
सारे दोस्त
सिर्फ आदमी का
थोड़ी देर स्क्रॉल करना है और चल आने तो जल्दी चल 2 मिनट ही है चलो चलो उसके बोलने नहीं हां बस देख 2 मिनट अंदर साइकिल का होना अभी ना
कोई आने
यहाँ की लगाते कैसे लगाओ या बंद करो तो लगी नहीं है खराब हो जाता है अरे वहाँ चलो यहाँ मैंने पूरा उठा दिया था ना मुश्किल कंप्यूटर ही नहीं था उनका ऐसा मानना था कि होता है लेकिन कुछ हुआ नहीं गर्मी लगती तो बस नहीं ठंडी जैसे कि हम कब से करेंगे इसका तो ऑन करना था ना अभी कैसे ऑफ मत करना टेंपरेचर इंक्रीज या डिक्रीज करना है बहुत गर्म हो रहा था उसमें नींद आ रही थी हाँ गर्म हो रहा था अभी टाइम ये है 9:15 तक है तो 9 और वो लोग भाग गए अभी बट मैम हमें ज्यादा वक्त दे दी शायद 9:04 हाँ मैम ने थोड़ा लेट हमारा थोड़ा कंप्लीट चला मैम अभी फैमिली क्लास के बीच में मुझे
You finished with the first question? Second? I gave you 5 or 6 questions, no? एक मिनट दिखाना वो. Now, so the first question we discussed, the second also we discussed. Now, 25, did I give you 25?
Yes.
Yes. So 25, what are the questions? Name the process, you know what to write. Define, you know what to write. When the weight of the test tube A and B is taken before and after the experiment, what is observed? What will you write?
The weight of the test tube A increases. The water level drops in test tube A.
When the weight of the test tube A and B is taken before and after the experiment, what is observed? Test tube A, test tube B. Test tube A will show remarkable decrease in the water level. The water level decrease in test tube B will be lesser. Did you write that? Did you write that? So always to remember, test tube A will show remarkable decrease. Test tube B, decrease in the water level will be less. Give reason to justify. Why in A we are saying water level decreases remarkably? Why? What reason you will write? Tanmay, what reason will you write? Did you write? Why there is remarkable decrease in water level of test tube A? Why? Oil is not there. So oil is not there. So transpiration and surface evaporation both will contribute to decrease in water level. Transpiration and surface evaporation. Please write down. This is what you have to write. Why did you write this?
I did 29, 30, 21st.
अच्छा 21 किया नहीं, 25 नहीं किया है अभी. I will tell you the answers. Are you writing it in your notebook?
Yes.
So page question number 25 का लिखो. Question number 25, 1, 2 you will write it on your own. You know that. Number 3 लिखो. When the weight of the test tube A and B is taken before and after the experiment, what is observed? You are straight away to write. Water level in A decreases remarkably, and in B, capital B, and in B, water level decreases negligibly or much lesser. Not right negligibly, right? Much lesser. The decrease in water level is much less. Okay. Reason. They have also asked reason. Next you write reason. Next line. This is because in test tube A, both transpiration and surface evaporation contribute to the drop in water level. Full stop. In test tube B, there is no transpiration. Oh, I'm sorry. In test tube B, there is no plant, no? So what I said, what is the observation? I'm sorry. When the weight of the test tube A and B taken. So I asked you to write, in test tube A, the drop in the water level is remarkable. And in test tube B, there is no drop in the water level. The water level remains same. There is no shoot, no. The water level remains same. Acha. Now reason. So for reason for A, you have written both transpiration and surface evaporation contribute to drop in the water level. For test tube B, which acts as a control, no transpiration, and also in control there is no transpiration because there is no shoot, no? There is no transpiration, and also the layer of oil prevents surface evaporation. So once you write 4 or 5 answers like this, you are acquainted also with the basic technical terms what to write in your answer, no? So remember the format of writing. So in test tube B, there is no transpiration and oil prevents surface evaporation. So that is one, right? What is the purpose of keeping the test tube B in the experimental setup? It acts as a control. Next, I asked you to do 29 and 30. 29 and 30. Karo pehle, then I'll discuss with you. 29 and 30. Selena, the rest 3 questions that I asked you to do, karo. Volume dikhana. From your volume, you can try these 2 questions. Selena, पहले वो complete करो. 3 questions that I gave you. वो पहले complete करो. And from your volume, all of you can take 73 page. Question 12. Page 73, question 12 and 15. This we will do later. Page 73 ka 12 and 15. 12 is from that book, from that setup. 12 and 15 I said.
Yeah.
72 बचा है ना? 2 questions left that I did not discuss with you. करो, try करो. Finish with 25. No, now next. वो करो. Then we'll give you the book again. What is mentioned? 5% sucrose. This side is 10% sucrose. The water will move from B to A. The level of water or level of solution in B will decrease, A will rise. Soham, any doubt, anything, any problem? Radhika, what happened to you? You're not well? What happened?
I have fever.
Asha, that was not Mr. COVID, no? Thank God. Again, she is feeling cold. Both of you feeling cold again? Huh?
I'm just wearing it.
That one, another one. It's winter, no? Feeling cold. Prashansa and Tanmay, they are hot. Very good. Warm-blooded. At this age, your blood should be boiling. क्या sweater पहन के? Icse a lot to study. Don't wait for the exam bells to ring. If you intend to get good marks, study regularly. Every day, unless and until you finish some portion of some subjects, unless and until you have a major problem at home or with your body, that should give you discomfort to go to bed at night. Huh? How many of you feel guilty? जिस दिन कुछ भी पढ़ाई नहीं हुआ and it's already night and you go to bed, do you feel guilty that आज मैंने कुछ भी नहीं पढ़ा, कुछ भी नहीं सीखा? Do you feel guilty? So that's sign of a very good student. वो बेचैनी होनी चाहिए. अरे आज तो मेरा पूरा दिन चला गया, waste. हाँ होती है. Yesterday only. अरे वाह. तो किस वजह से तुम्हारा पूरा दिन कल बर्बाद हुआ? That is okay. ऐसा नहीं कि तुम. At the same time, I am not telling you that make your life so boring and horrible. But you should know how to balance your life. चलो आज घूम के आए. Tomorrow you went out for a weekend. ऐसा कुछ है. Try to make it up. The other day when you are at home, try to make it up. For how many of you, your parents are after your life to study? Shame on you.
Ma'am, not like that.
Joanna also.
No, ma'am, not like that. But they always encourage me. They always
encourage. That is okay. But they should not be
not
like
like
stick
that
after
behind
you
me.
कि पढ़ो TV देख रहे हो मोबाइल पे हो.
No, ma'am.
That much responsibility अभी हो जाना चाहिए कि मेरा life है, तुम्हारा future है, तुम्हारा ही life है. If you don't study, you know what's going to happen. And if you study well, you know what's going to happen. You can fulfill all your dreams and wishes and desires only when you study till a particular level. उसके बाद hard work. Right now, more than hard work, it's your study that matters, no? So for a 9th and 10th standard student, this should be very clear. हाँ बोलो, doubt? एक बार book दिखाओ ना, please. What is it? What is it? अच्छा, name the process. Why was the pot enclosure rubber sheet? You know. Mention two external factors which can excel. Okay. External factors, light and temperature. Very good. Two adaptations. We are here to learn this. Two adaptations. Could you write? Two adaptations to reduce. You can write sunken stomata, reduced stomata. Okay. Rolling of leaves, shedding of leaves, rolling of leaves, fused stomata. Okay. Draw a neat label diagonal stomata label. Now no need to draw. When we learn that part of the chapter, that time only ask you to draw in the class. Abhi mat karo. Okay. Shedding of leaves, reduced stomata, rolling of leaves. Done. 7th day अभी है और कौन सा क्वेश्चन बाकी है? करो 29 करो. देखो कोई है क्या डाउट?
मैम ये टोटल 12 and 15
12 and 15 volume का. For you all, you don't have to do 12. The same thing that you have done in your book. 12 तुम्हारे book का repetition है. I gave you one question like that. देखो 12 में the one which is there in the volume is the same repetition that you have done in your book right now.
So we don't have to do that.
हाँ. 12 and 15 not for 7th day. The same thing that you have done in your book. Huh? From volume only for people who are using Selena 12 and 15. Because they have more questions in their books. Okay, 7th day, if you have finished that, you will go to the sample papers. You don't have sample papers. So share the, take the Selena from somebody. And वहाँ के sample papers 3 solve करो and finish up. वहाँ पे you may not write all the answers in the notebook. Only just go through. Do you know the answers? Just check it out. Just check. These are previous concepts that you have studied. Share करो तीनों एक साथ करो. एक.
I completed 21. Can you discuss?
So now 29 and okay 29 and this those of you did that volume question number 12 for 7th day. Check answers of 29 and for volume I asked you all to do number 12. Please check that first. Volume question number 12 that I asked you to do now. The same hai. I'm discussing this answer. Please check. Volume and this is same. Now 7th day, name the physiological process. Check what you have written. 29.
Transpiration.
Why was oil added? Surface evaporation. When placed in bright sunlight for 4 hours, what do you observe with regard to the initial and final weight of the plant? The weight of the plant will decrease due to transpiration. Suitable reason for your answer? Acha, wait. What do you observe with regard to the initial and final weight of the plant? Okay, name. In the first part of the answer, you are not supposed to write transpiration because this is what you are supposed to write in the next part. So, jo 2 hai, sub number 2. Check your answer. Write it the proper way. What do you observe with regard to the initial and final weight of the plant? there is a drop in the weight of the plant. There is a drop in the weight of the plant after 4 hours. There is a drop in the weight of the plant after 4 hours. Give a suitable reason. That's the next part. Give a suitable reason. The drop in the weight is due to the loss of water by transpiration. The drop in the weight is due to loss of water by transpiration. 3. What happens to the level of water when the setup is placed in humid condition? There will be a negligible drop, slight drop, negligible or slight drop. There will be a negligible drop. Because they are talking about 4 hours. 4 hours में negligible drop होगा. There will be a negligible drop in weight in 4 hours. You are talking about 4 hours, remember. वैसे तो अगर time नहीं mention है, then the other way of writing is the drop in the weight will be very slow. But we are not going to wait for 12 hours now. We are going to wait only for 4 hours. So 4 hours में there will be a negligible drop in the weight. Windy condition, the drop in the weight will be comparatively faster. Comparatively word लिखना है. The drop in the weight will be comparatively faster. Mention any 3 adaptation. Narrow leaves, loss of leaves, fused stomata, shedding of leaves, needle-shaped leaves. So, thick cuticle, ये सब भी आता है. Thick cuticle, okay. Explain the term guttation. Do you remember?
Loss
of water by the hydrothodes in the form of water droplet. Water droplet. Loss of water by hydrothodes in the form of water droplet. 30 और 15 जो मैंने दिया है वॉल्यूम का सेम है. 30 and 15 of the volume that I asked you to do, no, 12 and 15 I asked you to do, no, from the volume. 30 and 15 is same. Now, Sadhu Baswani will set up questions are over, this side also they are over. And volume question is a repetition of what they have, 12 and 15. Only for Selena you can do it separately. Sadhu Baswani no need, this 7th day no need of doing it separately. Now, just finish with the 3 questions of Selena that is there in the sample paper. I want all of you, those of you who are not using this book, share and do it. Just go through and find out if you know the answer. Notebook में लिखने की ज़रूरत नहीं है. Those of you are not having Selena, ठीक है? Those of you are having Selena, you may write it down in your notebook. या तो textbook में भी लिख सकते हो. And those of you are not using Selena, just go through and find out whether you know the answers. Doubt या confusion है, clarify करो, and then I want to proceed ahead. ये वाले आंसर्स if you want you can write down in the book itself. लिखना है वहाँ पे लिख दो.
Now we have a question here. What is meant by DNA duplication which occurs during interphase change? So chromosomes are duplicated and also
Duplication of DNA. What is meant by DNA replication? Duplication of DNA.
DNA.
Doubling of DNA. Duplication of DNA. Only that. Duplication of DNA.
So DNA replication means duplication.
Duplication of DNA, that is formation of an identical copy of DNA. That much, one sentence में इतना ही लिखना है. Duplication of DNA, that is formation of an identical copy of DNA. Formation of an identical copy of DNA. Any doubt? हो गया सारा? All done? ये कौन सा क्वेश्चन है? नंबर क्या है? 7. Selena sample paper का नंबर 7. Is it clear all of you? नंबर 7 जो मैंने दिया है सॉल्व करने के लिए 3 क्वेश्चन देखो 1 by 1 करो फटाफट करो हम मेक इट फास्ट another 5 minutes I'll give you finish up Done, all of you? Is it out there? Any doubt in the question?
Yes, ma'am.
Bolo. Bolo.
X chromosomes.
Huh? Homologous chromosomes. Now write down the definition. If anybody else also has the same, please write down. Homologous chromosomes, I explained you know in meiosis. A pair of chromosome which has one maternal and one paternal chromosome. हाँ, parental तो सभी parental है. One maternal and one paternal. A pair of chromosomes which has one paternal and one maternal chromosome is a homologous pair of chromosomes. Is a homologous pair of chromosomes. Done now? All of you done? Okay, it's okay. Done, all of you done? All questions done? No, this is fine. Okay. Now what are we supposed to learn? We are moving into the next part of the chapter, and now we are trying to understand transpiration by the photometer method. Transpiration by the photometer method. There are various different types of photometer. What you are studying in the chapter right now is Genong's photometer. You are studying Genong's photometer. What is photometer? It is a device. It is a device, basically a glass device. It is a glass apparatus. It is, and in this glass apparatus, we come across that in this glass apparatus, the arrangement of the parts is as such which can allow the movement of the water, movement of liquids. So now, when we are talking about that, what does this setup include? This setup majorly includes a funnel-shaped part of the apparatus where water is filled, and this funnel-shaped part is continued as a bent glass tube. As a bent glass tube, water should be there in this bent glass tube, or water is there in this bent glass tube. The other end of the glass tube should directly be in contact with some colored water. It should be connected with some colored water. So what are we taking in the beaker? In the beaker, we are taking colored water. We will learn why. In the beaker, we are taking colored water. And there is a reservoir. Reservoir. You know what is the meaning of the word reservoir? Reservoir means where there will be adequate water placed, and from time to time that water will be allowed to drain into this glass tube. The water will be allowed to drain into this glass tube by this. So now when we are talking about this entire setup, the entire photometer, what does it contain? It majorly contains a very long, broad glass tube, and at one end of the glass tube, there is a funnel-shaped structure. There's a funnel. We are supposed to place a shoot in the funnel, taking care that this stem region, if you remember, I already told you that whenever we cut this stem, we cut the stem this way. Meaning, I'm not supposed to cut the stem this way. There's a reason. That shoot, when I take, I am not supposed to cut it this way. I am supposed to slash it, and then this type of shoot, I'm supposed to place it here. Why? Because if I talk about this oblique cut of the shoot, the stem region, the oblique cut, it is understood that the number of cells exposed to the water is more as compared to this. So I'm talking about whether the surface is this way or the surface is this way. Does it make a difference? It makes. The number of cells exposed in this type of a cut is always more. So I don't know if at all you have noticed that these gardeners, when they plant the stem or the saplings, in the soil, if at all you have noticed, they would always hold the plant and always they would slash it. They don't chop it this way. They slash it this way. Believing that more the number of cells are exposed, more would be the absorption of water. Of course, here the number of cells exposed or exposed to the soil water is less. Here it is more. Okay. So this is what we believe here. That what are some of the precautions here now that we take? Number one, the stem cutting should be in oblique. The stem should be obliquely cut to allow maximum number of cells exposed to water, in contact with water. Next is what? We are talking about this reservoir filled with water, and this knob you can regulate off and on to allow the water move this side. Then what else? This is a capillary tube. This is a capillary tube. If you remember the concept of capillarity, you have learned in absorption. What is the concept we have learned? For this capillary tube, the moment I dip this end of the tube in the colored water, is it not? Small amount of colored water will as it is rise in the capillary tube? Yes. These capillary tubes are supposed to be very narrow. And narrow the capillary tube, more is the suction force. So the moment we dip the capillary tube, the entire glass tube, the glass tube has got an outer lining, outer layer, outer covering I should say, and inside there is a very narrow capillary tube. The moment you dip the glass tube in this water, small amount of liquid as it is goes into the capillary tube by capillary force. The same concept of capillarity that we have learned earlier. Yes, but is our objective to draw the colored liquid only? No, our objective is not only to draw the colored liquid in the tube. Our objective is something else. Now, what else are we trying to study? Now, on this glass tube, there also should be a calibrated scale, a graduated scale should be there. A calibrated scale should be there, which should have some readings. Something like 1 and then 2 and then 3. So it is something this way. So we are talking about a scale which generally is run till 10. Then, okay, so this is more or less a setup. The glass tube, the capillary tube, and then the graduated scale, the calibrated scale is there. The calibrated scale is there. And then, when we are talking about what is the need of the calibrated scale, we are going to read something here with the help of the calibrated scale. Let us see what. The first step that we try doing with this setup is, we try to, this entire thing is more or less fixed. You cannot move this glass tube once you have put it with the help of a clamp stand. It is there. With the help of, it should be, the entire setup should be with the help of a clamp and stand. Once you have fixed this, you cannot move this tube, this bent glass, you cannot move. So what I'm going to do, we are supposed to take this colored liquid out and in, like this. But what are we supposed to introduce? What are we supposed to, what are we trying to enter? What are we trying to draw inside this capillary tube? We are trying to draw a specific amount of this liquid. Apart from the liquid, we are trying to draw a bubble, a single bubble into this capillary tube. That's the most difficult part of this experiment. Now this setup looks very easy, but when you try to do it in the laboratory, it's very difficult. Why? Because as I'm taking this beaker out, this entire beaker has got colored solution. I take it out and again I place it in. I take it out and again I place it. The moment I place it in, there are chances that a lot of bubbles will start entering the capillary tube. Do we want that? No, we don't want. We want only one bubble to enter. We want only one bubble to enter, and that's the most difficult part of this experiment. So we have to take out, displace this beaker and place it back, displace it and place it back. And that has to be done a number of times. And then maybe now we are able to get one bubble in. So it is something like this. This is the mouth of the capillary tube. Is the colored solution entering into this tube? No. The colored solution is not entering into this tube. The colored solution is entering only into this middle tube, the capillary tube. It is entering only into this capillary tube because the other two tubes are not open, it's closed, which will be just there. So now when we are talking about the water, so what will happen there? As I take this beaker in, what's happening? Yes, small amount of the solution will move into, and we try to make it a point that only one single bubble enters. See now, we are able to introduce only one single bubble. Now next what? We allow the colored liquid to keep flowing. As the colored liquid keeps flowing, our bubble also keeps moving. The colored liquid keeps flowing, the bubble also starts moving. The bubble is also moving. Now when the bubble is moving, what do we try to understand when the bubble moves? I'm sorry. This capillary tube and reservoir should be connected. So now, when we are talking about this, what I told you, that now a single bubble. Can you see this single bubble that I have drawn here? Only one single bubble. One single bubble we are able to introduce. So now next is what? Yes, the colored solution is moving in. As the colored solution moves in, the bubble also moves. Maybe now, after some time, this single bubble has moved up to here, or else the single bubble has moved up to here. Now as the single bubble has moved up to here, are we supposed to start our experiment? Yes, we are supposed to start our experiment this way. Now what are we waiting for? We are trying to find out that this single bubble should actually try reaching the zero mark. I'll draw the capillary tube a bit broader so that you understand. So now this is what the capillary tube is on the either side. So what I was saying, we are capable of drawing only one bubble, but we have to wait till the bubble reaches the zero mark. That's the start of our experimental time. So now the bubble has reached here. The bubble is no more there, because once the bubble has started moving and the liquid has started drawing in, there will be no more bubble introduced. So now we are not supposed to very vigorously take in and take out the beaker. That's a very important thing that we do. Very, very cautiously. very slowly take it very close to the mouth of the glass tube so that a single bubble gets in. Now when a single bubble gets in, we are starting with the experimental period of time. Now maybe from this time, we wait for almost 1 hour and try to see how long the bubble moves. Right now we are at the 0 mark. We now try to see how far the bubble moves. Maybe in 1 hour time, the bubble moves till here. Are you expecting that the bubble will move very fast? No, the bubble will not move very fast. The bubble has moved from 0. Initially it was at 0. In 1 hour, where did it move? In 1 hour, it has moved till the 1 mark. What is the reason? Why did it move till the 1 mark? Why did the bubble move forward? The bubble moved forward because this colored liquid which is there, this colored liquid is all getting mixed here, no? So this colored liquid which is moving this side, the colored liquid which is moving this side is also dragging the bubble, allowing the bubble to move. But then what is the force which is allowing the liquid and the bubble to move? The loss of water. The loss of water from the leaf. What is allowing the bubble and the water to move? The loss of water from the plant. And since there is loss of water from the plant, there is absorption by the way of the stem cells. By the way of the stem cells. So these two processes are happening side by side. What is happening there? First of all, for a very short period of time, that concept of capillarity is going to work. After that, can I say that it is the capillary force which is dragging the water and the bubble? No. If only capillary force is there, then the liquid will stop here. The bubble will stop here. It will not move further. But what is the reason that it is moving further? And gradually it is moving very slowly. But it is moving. Why? Because the leaves are constantly losing water. The leaves are losing water and the stem cells are also absorbing water. By the way of the xylem, the water is moving. So by the way of the xylem, the water moves. By the way of the xylem, the water moves and the water goes to these cells. The water goes to these cells and then the water goes up. Is this process happening very vigorously? No, very slowly. Had this been roots here, then the process would have run much faster. But we don't want the roots because the setup is so small. We don't want roots. So then now next, what? Okay, for 1 hour we saw that the bubble has moved till the 1 mark. But in this type of experiments, we cannot only rely on one reading. We need to, because environmental conditions are changing, and along with environmental conditions, the movement of the bubble also will vary. So in the first hour, the bubble moves till 1 mark. Now I want to put back the bubble into the 0 mark again. I want to take the next reading. Second, same movement of the bubble I want to see for the second time. So for that, am I going to start from 1 and see further? No, I want to put it back to 0. How will I put it back to 0? Meaning the liquid should move in this direction now. This liquid in the capillary tube should move in this direction. तभी जाके bubble back 0 में आएगा. That's a very delicate thing that we do. That's the reason very difficult this is. But इसके लिए अभी क्या करना है? ये reservoir open करेंगे. जैसे ही reservoir को open करेंगे, small amount of water will start flowing in. And small amount of water flow in करेंगे, and then this bubble, and then ये ध्यान रखना है, the bubble again should not go down. Generally the tendency जैसे हमको फटाफट अंदर चला जाता है, and again you have to start from 1, again start. Again first reading चला गया, first reading से तुमको फिर से लेना है. नहीं, हमारा first reading हो चुका है. First reading में 1 तक आया था. Abhi, next what I want to do, open the reservoir in such a way very slowly so that put the bubble back into the 0 mark. यहाँ तक आते ही बंद कर देना है. Put the bubble back into the 0 mark and close it again. Now again, check for the next 1 hour. Check for the next 1 hour. Ab next 1 hour में bubble, how far does it move? That is what we are trying to understand. अभी शायद environmental condition थोड़ा change तो हुआ ही होगा पिछले 1 घंटे में. Now that I talk about 9 to 10 o'clock, whatever the environmental condition was, or is, can we predict that from 11 to 12 o'clock it's going to be the same? Or 7 to 8 o'clock, was it the same? No. So यही हो रहा है यहाँ पे भी. So now in the next hour, the movement of the bubble may be slow, may be fast. दोनों हो सकता है. So in this experiment, maybe I believe that पहले 1 hour में the movement was something like till the 1 mark. Now let's see what. Now in the next 1 hour, we get to see that the bubble is comparatively moving much faster. And the bubble has moved almost till the 1.5 mark now. The bubble was moving no in this direction, the bubble is no more here, and the bubble has moved almost till the 1.5 mark. So now, when we are talking about that it has moved from 1 pele, now 1.5, duration is same. Going 1 hour we are testing, 2 hours we are testing, whatever we decided. So इस तरह से 3 readings minimum लेना है. And then we generally for any type of this biochemistry or chemistry experiments we generally take base average of 3 readings. So इसी तरह से again I want to take the 3rd reading. So what will I do now?
Open the
Bubble को 0 तक लाना है. That's the start point. तो bubble को 0 तक कैसे लाएंगे? How to do that? Open the reservoir. थोड़ा सा पानी, this liquid from the water from the reservoir try to drain in the opposite direction, then only the bubble will be pushed backward. The bubble will be pushed backward. And now when the bubble is pushed backward, we understand, chalo, bubble again has reached the 0 mark. Little bit more force, bubble will come down. That again we'll have to keep a check. Again here. Now the next 1 hour we want to check. And now maybe we find that the bubble movement again has become very slow. And maybe now in this the bubble movement has come much lesser than 1, or it is also possible that it is not exactly 1.5, but it is something like 1.2 or something. It is something like 1.2. So what has happened? In the 3rd hour it is something around 1.2. Now take an average of this. So ultimately what are we understanding? We are checking this bubble movement 0 से कहाँ तक गया? 0 to 1, 0 to 1.5, 0 to 1.2. It can be 0 to 2, 0 to 3, 0 to 2.5, anything. Whatever the duration of time we decide. Now ultimately what are we trying to study with this bubble movement? What are we trying to study? We are trying to study that in this durations of time, the bubble is moving from one point to the other. Ultimately, we are trying to understand that what is the reason why the bubble is moving forward. The reason only and only is the loss of water by the leaves of the plant. This loss of water from the leaves of the plant is responsible for a particular pressure at this region. This transpiration, the higher rate of transpiration, higher will be the absorption at this point. Now here, with reference to the photometer itself, let me just tell you one more concept here. What is known as transpirational pull? What is known as transpirational pull? Now this is my plant. And whenever I talk about the suction force, now this is one of the significance. The significance concept with relation to this, try to understand. So now here, I'm saying that this is the xylem tube, which is now connected with the veins and the midrib of the leaf. And then this is my leaf here. And these are the surrounding cells of the leaf. Now, in this case, what are we supposed to understand? What is the meaning of suction force? What is the meaning of suction force? And what are we trying to understand as transpirational pull? What are we trying to understand as this definition? Transpirational pull is the suction force which is developed at the level of the roots, due to which there is adequate process of diffusion of water and its upward migration. Transpirational pull is a suction force which is generated due to what process? The rate of transpiration will decide how much the force should be here. Whatever is the rate of transpiration, that much will be the rate of osmosis here. And they are almost at par. Almost at par. यहाँ से जितना गया, वहाँ से उतना absorb भी होना चाहिए. So, when we are talking about transpirational pull with reference to absorption by roots, also I had explained you a little bit of that. That what is exactly happening? These are all group of cells. These are all group of cells. No, these are the xylem cells. And now, these root cells, what will happen? The water enters and the water enters and the water enters and the water starts moving up. So यहाँ पे this process which is known as osmosis, which already I have studied in a lot of detail. So now we are trying to only understand that why the rate of transpiration or how the rate of transpiration can control the process here. If you remember, we already studied that higher the rate of transpiration, there is always a vacuum created. If this is what the xylem I understand. And do you know why the vacuum is created? Do you still remember why I said the vacuum is created?
Because of force of coercion between.
Because the water molecules always, they never remain scattered in single molecules, but always in a chain. They are always in a chain. They are always in a chain. What is that chain called? Or what is that property called? Cohesion. Water molecules are always in a chain. They are released. 8, maybe 10 molecules are in a chain. So all this 10 will get released at one go. एक करके नहीं. अगर एक करके जाता, then the entire tube would have been filled with water molecules. Then the pressure also would have been much lesser. But it's not like that. 10 molecules at one go will get out. 5 molecules at one go will get out. 15 molecules at one go they will get out. It's like that. So first of all, वो property याद रखना है. So now if this is a trail of water which is entering, so all these will go out at one go. So ultimately this space is empty. So पहले तो this space will be filled up by another few molecules जो trail में है, filling up. Then this space is empty, so this space, so this already you know that this vacuum is filled up. तो ये vacuum जब यहाँ तक पहुँचेगा, the vacuum will be bigger on a humid day or on a bright sunny day. On a bright sunny day, this vacuum will always be formed, and very fast this vacuum will be formed. Every time the vacuum will be formed. So on a bright sunny day, ये vacuum बहुत जल्दी बनता रहेगा, है ना? Every time it will get formed. And that's the reason the absorption also will be much faster. The osmosis also will be much faster. So ये process is almost same like what we are trying to understand here. More the loss, more is the absorption. But यहाँ पे we are not talking about root, तो इसीलिए absorption is comparatively slow. But यहाँ पे तो हम real situation we are trying to understand where the plant already has got a spread out root system. So obviously osmosis is much higher. So one of the significance that you talk about so far transpiration is concerned is generation of suction force. Yet written also in one of the answers that we discussed, that one of the significance, this is a major significance of transpiration. This is a major significance of transpiration. और इसके आगे पीछे जो भी हम significance consider करेंगे, this is related to this only. अच्छा, कैसे? So first है ये जो transpirational pull है और suction force है, because of this the water is constantly moving, water is continuously moving, the water is continuously moving. So यही लगभग what we are studying in this experimental setup. Okay, now the suction force, water is continuously moving. तो ये water continuous movement की वजह से what is the plant or how is the plant also otherwise benefited? The plant is also otherwise benefited because इसी process की वजह से distribution of water तो हो ही रहा है plus distribution of minerals भी तो हो रहा है because the water is not only water, it is water where large number of minerals and ions are dissolved. So this is osmosis, a continuous process. This is because of transpiration, and this suction force which is generated because of the rate of transpiration is transpirational pull. So what is the definition of transpirational pull? The suction force which is generated in the xylem tubes or in the xylem cells. Suction tube which is generated at the xylem cells of the root due to continuous transpiration. Likho, what is the basic definition of transpiration? Transpirational pull, the suction force which is developed. Suction force which is developed at the xylem cells of the root. Developed at the xylem cells of the root due to continuous transpiration. Due to continuous transpiration, which allows the continuous uptake of water and cell-to-cell osmosis. Which allows continuous uptake of water and cell-to-cell osmosis. So, transpirational pull is always associated with the rate. Is the transpirational pull affected by environmental factors? Yes. I gave you 2 instances. So, अभी कौन-कौन से environmental factors से majorly it affects? Temperature, sunlight, wind velocity. Now, say suppose a wind velocity Q. Now, this is a leaf. There is adequate amount of water vapor. If the temperature is high, intensity of sunlight is high, this water vapor is constantly getting dried out. So obviously, this water vapor is getting dried out. मतलब ये atmospheric area जब is more or less dry. This atmospheric area, meaning the atmospheric air surrounding the leaves, when they are dry, the leaves will release more water vapor. On a windy day, this water vapor is blown away. So, फिर से, the surface or the atmospheric area or the atmosphere surrounding the leaves is still getting dry. So, either on a bright sunny day, when the temperature is adequate, light intensity is adequate, or on a windy day. Parameters are 2 different I'm talking about, but ultimately same thing is happening surrounding the leaves. क्या हो रहा है? The surrounding of the leaves are becoming dry. The adjoining regions, leaf के आसपास, the atmosphere is dry. Leaf के आसपास, the air is dry. So if the air is dry, the leaf will be in a position to keep away more and more water vapor. But on a rainy day, what is happening? Whatever amount of water vapor the leaves have given out, it is there for a long time. The water vapor surrounding the leaf is saturated. Because now there are strong winds to blow it. And even if there are strong winds, the surrounding atmospheric region all are saturated with water vapor. तो वो water vapor जाएगा भी तो कहाँ जाएगा? The next region is also saturated with water vapor. The next region is also saturated with water vapor. All around there is water vapor. On a monsoon day, on a rainy day. So on a bright sunny day, this water vapor is not accumulating near the leaf. They are just getting moved away. So the surface or the surrounding areas of the leaf are always dry. The leaf gets adequate amount of space to release the water vapor. But on a humid day, on a monsoon day, on a rainy day, condition is just the opposite. So on a bright sunny day, जैसे ये process ज़्यादा fast होगा, the vacuum created is much faster and this is much faster. The absorption is again much faster. So ऐसे जैसे ये faster हो रहा है, it's not only the transport of the water, but also the water and distribution of the minerals. Now, if I talk about the other point, what is the other point that we know? Cooling effect. Cooling effect, matlab? Why is it very common a feature? If I talk about, why do we generally on a summer time, no? Why do we generally tend to stand under the big trees, under the shade of the trees? No, when we are feeling very hot. Say suppose when we are traveling, also we are going for some. So we always have a tendency to shade me under the plant. Let us sit down for some time. Why is it soothing to stand or sit down or sleep under the tree? Why? Because there is always a cool breeze. Why that cool breeze? Why that cool air? Because the leaves are constantly releasing water vapor. The leaves are constantly releasing water vapor and making that surrounding area a little bit cool, making the surrounding area a little bit cool. But is the plant so much thoughtful about us and our benefit? No, the plant is also thinks about itself. The plant is also enjoying that benefit because whenever there is adequate amount of water loss in the form of vapor, yes, the surrounding air has got a lower temperature. This particular air will have a lower temperature than the surrounding air, and that's the reason the cooling effect. But that cooling effect, the plant is not only thinking about us. Remember that animals ko cool karna hai. No, the plant is actually bothered about itself. This cooling effect is because, or this cooling effect is also affecting the plant cell cytoplasm majorly, so that the cytoplasm में जितने सारे enzymes है, if they are heated up, enzymes are majorly proteins and they'll start breaking off, and the functional activity of those enzymes and pigments will be lost. Even chlorophyll, if I talk, chlorophyll at a very high temperature, it gets denatured, broken down, and even at a very low temperature, it cannot show its function. So now that I talk about cooling effect, ये खाली ये नहीं समझना है कि the plant is releasing water vapor to cool us. Not that. The process of cooling effect, or why do we say it's cooling effect? Cooling effect for the plant itself, so that the enzymes and the pigments that the plant has in its cytoplasm should not be subjected to a very high temperature, and if at all, the enzymes and pigments start becoming non-functional. Their configuration is lost, broken down, bonds are broken, etc., etc. So cooling effects, of course, we are benefited, that is true, but the plants are also benefited. So their protoplasm, their cytoplasm remains in a functional state. All the enzymes and the proteins remain in a functional state. So ये तीन हो गया significance of transpiration. You are supposed to understand this, a major function to understand, a major property that you have to understand. Distribution of water and minerals, you know which type of tissues to name here, and the cooling effect just now that I mentioned. Then for photometer, now let's go back to the experimental concept again. For the photometer, what did we learn? We learned that with the reference of movement of the water molecule, the water bubble, we are more or less trying to understand how much of the water lost. So, what does the photometer generally signify? What can we measure by the photometer? We can measure the rate of water intake. The rate of water intake. This is what we are trying to measure. At this region, what is happening? This is what we are trying to measure. The rate of water intake. यहाँ पे क्या हो रहा है? That is what we are trying to understand. Is this clear? But I already have been telling you, repeatedly I have been telling you, ये rate पे ये rate dependent है. So this is more or less at par. Whatever is the amount of water loss, the amount of water intake is more or less the same. Water loss and water intake is more or less the same. Roots hota to process faster hota. Now we are not talking about roots. So what does this particular apparatus, the photometer, try to measure? You are not to write it measures the rate of transpiration. That is not a very appropriate answer. क्योंकि क्या हम rate of transpiration यहाँ तक ही limited है? No, we are not limited only till here. We want to understand how much of the water is going through the way of the stem, because इससे हमारा movement of the bubble dependent है. तो हम तो यही observe कर रहे हैं ना? This is our observation, no? Our observation is this. Is our observation whether the water level dropping or not? No, that is not our observation. हमारा observation है water bubble कितना move होता है in 1 hour time, 2 hours time, 5 hours time. So water bubble movement is dependent on what? Rate of, no, I'm sorry, water bubble movement is dependent on the rate of water intake, which is again dependent on rate of water loss. So you are asked questions. What does the photometer measure? Rate of water intake by the plants. Rate of transpiration नहीं लिखना है. अगर हम यहाँ पे colored water के जगह, if you take just water, then
The air bubble won't be visible.
the air bubble won't be visible क्योंकि दोनों side में glass tube है. This is also a glass tube, no? So what I said, this is also a broad glass tube. This is also a broad glass tube, and this is also the other glass tube. So ये एक glass tube है, ये भी. So in between the broad glass tube, there is a narrow capillary tube. तो ये narrow, ये तो already transparent है. The outer margins of the glass tube, upper and lower, the outer regions of the glass tube, as it is transparent है ना, this, if you see this one, the outer regions of the glass tube, ये जो blue region दिखाया है, this is the glass tube. So in this blue region, if you see the outer white region, that's a glass tube. And inside there is a capillary tube. Can you see the blue region is the capillary tube? So ये outside region तो white है, transparent है. And if you take water instead of a colored solution, then this also becomes white. Will it be easier for us to understand the movement of the bubble? No, it will be difficult. So that's why we take a colored solution, blue, red, pink, whatever. We take a colored solution. So as the colored solution moves, we can see something transparent moving. The outer walls are transparent, only the central tube is colored. And then the movement of the bubble we can understand very clearly. And then we have to keep marking the first reading, the second reading, the third reading. What are some of the limitations? Now, as I already have been mentioning that this apparatus looks simple, but when we try working it out in the laboratory, it's difficult because ये जो fineness है, bubble को बार-बार zero तक ले जाना, and then again starting it, and then again, and then introducing that one single bubble is the toughest job in this experiment. It's very easy to introduce 5 bubbles because the moment you take out this beaker and place it back, बहुत सारा bubbles will be getting formed and they'll start entering. No, we don't want. Reservoir open करो, again put all down, again take it out, again. So ये process many times we have to do. Many times we have to do. But now say suppose we are successful in allowing only one bubble enter. Now that is how we start our experiment. But there is something more that we have to look into. Can we allow this setup remain for 3-4 days? Probably not. Q, because these shoot does not have roots. So will it remain alive for a long time? It will not remain alive for a long time. Or अगर ये alive नहीं है, the entire setup we have to change. We cannot carry on. The movement of the bubble, the dragging of the water, everything depends on the loss of water by the leaf. No? So what are some of the limitations you are asked in your paper? Limitations मतलब कौन-कौन से reason के लिए the working of this experiment में stop? Limitations क्या है? Number 1, ये जो working of this setup, this is very much dependent on the environmental factors. As I already said when I was explaining also, first reading may go very fast, second reading may slow down also. क्यों? Maybe in the first hour it was bright sunny, अब second hour में थोड़ा cloudy हो गया है. होता है. So this may differ. So first limitation is this process or the working is very much dependent on environmental factors. Number 2, the twig, the shoot may not remain alive for a long time or will not remain alive for a long time because roots नहीं है ना इसमें. Roots नहीं है इसमें. And सबसे important limitation or सबसे difficult जो part है, number 3, introduction of a single bubble. Introduction of a single bubble, that's very difficult. Okay, so these are the 3 limitations that you are supposed to understand.
The temperature has to be constant.
As long as we are talking about this, temperature fluctuation will happen, no? In this entire experimental period of time, say suppose I have to take 3 readings and I take the experimental time to be for 1.5 hours. So 3 readings मुझे लेना है, तो something around 5 hours I have to roughly wait. So 5 hours में temperature fluctuate होगा ही. We cannot keep the temperature constant. कैसे रखेंगे temperature constant? Temperature fluctuate होगा. Along with temperature fluctuation, just now what I explained to you, the surrounding atmospheric region of the leaf will vary, no? So उससे rate of transpiration भी vary होगा. So that's the reason the dragging and the movement of the bubble also varies. First reading may sometimes slow, second reading may sometimes much faster, third reading may sometimes again slower. Same reading kabhi bhi हमें नहीं मिलेगा. So this is what you should understand for the concept of photometer. Now you will do volume page question number 3. करो अभी. 73 question number 13. 73 question number 13. फटाफट complete करो. 73 question number 13. Question number 13. 73 question number 13 I asked you to do. 73 question number 13 fast. Textbook questions हम बाद में देखेंगे. First what I explained you just now, try to apply. Let me see the text. Selena, page 66. Question number 2 remains as homework. Selena, page 66. Question number 2 remains as homework. Same also? Yes. Let me check. For 7th day, page 4.15, page 4.15, 27 remains as homework. Same now, exactly same. अच्छा check करो. Same है तो repeat नहीं करना है. If it is same, so there is no need of same. Check.
We have a doubt. Which phenomena is demonstrated by this apparatus?
Which phenomena is demonstrated by this apparatus? Phenomena, you have to write rate of water intake. Rate of water intake. Process of or absorption of water by the stem also. That also you can write. Absorption of water by stem. Instead of rate of water intake, you write absorption of water by stem. Absorption of water by stem. Which process? Transpiration. Which process if transpiration? Done?
How should we explain absorption of water?
So they are talking about explaining also. Then in that case, actually they are asking for transpiration only.
So the definition.
Then in that explaining, you are not supposed to explain absorption right now here.
Yes.
Then in the first you have to write transpiration only. Transpiration and just the definition of transpiration. Definition.
And definition for explaining.
Explaining when you get, you are only to write down the definition, but little bit in detail. The loss of water by the aerial parts of the plant in the form of water vapor is known as transpiration. Full definition you have to write. That is explanation, 1 mark. But you don't have to explain in detail anything. Phenomena also they are talking about as transpiration. Set of questions same in your book also? Yes, questions are same, no? Okay. Done? All done? Done, so. So today we have learned about the experimental basis. Most of the experiments were already covered about the concept of transpiration. How many experiments we had learned today?
3.
Polythene sheet, 1 experiment. Bell jar, 2 experiment. Then cobalt chloride, 1. Weighing method, 2. Bottle wala experiment and photometer experiment. So all these experiments explain the same process of transpiration in different aspects. Process of transpiration in different aspects. Anything that you want to clarify? Complete karo pehle answer. Anything to clarify? So the first part of the chapter, experimental basis, all done. And along with that, also I'll explain to you the significance part. The next class we'll have to learn the process now. You have to learn the process. Arey Soham, why everything is falling down? Today only 3 hours lecture, no? Yes, enjoy. Arey see, सबसे ज़्यादा खुश तो क्यों अभी कहीं जाना है तीनों मिलके? नहीं? So on holiday, no meeting up with friends? हाँ अरे मैम ये तो कॉमन है ये क्या नया वर्ड है हाँ बॉयज नो बॉयज का कोई गेट टुगेदर नहीं होता है हॉलीडेज में क्यों पेरेंट्स अलाउ नहीं करते कि तुमको नहीं जाना है तुमको ही नहीं जाना वी डोंट प्लान हाँ जोना जाते हो not much
Parents don't allow
much.
They go in the evening.
Go in the evening. Garden, huh? That's also good.
This 6th one,
what is that?
Name the structure in the plant through which the above process takes place.
Name the structure. कौन सा ये transpiration? No, that's related to transpiration. Stomata. Primary structure is stomata. Name the structure in the plant through which the process takes place. Stomata. Structures है क्या? Structures. So stomata lenticels and cuticle. तीनों लिखना है.
The easy part of the line.
Structures. Parts में नहीं पूछा है. Structures. Structures अगर है तो stomata, lenticels, cuticle. Parts of the plant अगर पूछा है तो leaves and
The importance of the air bubble in the experiment.
name the importance of
Air bubble in the experiment. What is the importance of air bubble in the experiment?
what is the हाँ वही तो मैं सोच name the importance कैसे होगा what is the important तन्मय बहुत एक्साइटेड है आज 3 घंटे का लेक्चर है इतना दिन के बाद अभी तो आधा घंटा तो श्योर यहाँ पे जाने वाला है हाँ बाहर स्टेप्स के पास आधा घंटा मैं अभी रुकी अभी आधा घंटे बाद में निकलने वाली देखते now what is the importance of the air bubble the movement of the air bubble is proportional to the amount of water intake movement of the air bubble in the capillary tube movement of the air bubble in the capillary tube is directly proportional to the amount of water intake by the stem you should love to study you should love some sport also at the same time something that makes you relax you should chill with your friends also but friends के साथ हमेशा healthy discussion things to learn and share have a nice time all of you ये routine का book है ना हाँ
मतलब कोपर खेलने नहीं इन ऑर्बिट पे गए हैं इन ऑर्बिट पे गए